Okay, everyone, welcome to our question and answer on the natural history study. We'll get started. I'm sure people will join us as we go. My name's Chris Pearce and I'm part of SCN2A Australia, and I'd like to introduce Dr. Katherine Howell, who's going to answer some questions today on the natural history study. So Dr. Howell is a paediatric neurologist and hepatologist at the Royal Children's Hospital and is a clinician scientist fellow at the Murdoch Children's Research Institute. She's an honorary senior fellow at the University of Melbourne and a senior research fellow at the Florey Institute of Neuroscience and Mental Health. Her clinical and research work focuses on severe early life epilepsies and is supported by the National Health and Medical Research Council. In terms of her work on SCN2A related disorders, this has been important in describing their clinical features, determining a number of subgroups or phenotypes, and understanding the relationship between SCN2A phenotype and the impact of the mutation on the brain cell function. Dr. Howell is the leading investigator on the international natural history study of SCN2A related disorders, which will provide critical information for future treatment trials. So I'll hand over to Katherine now. The way we're just going to manage this is we're going to ask Dr. Howell questions that have been submitted. If you have any further questions, pop them in the chat and we'll address them if we can. Otherwise, we will get to them after this webinar. Thank you for joining us.
Thank you, Chris. And thank you, Dave.
Thanks very much for all the work that you're doing, Katherine, with the natural history study. It's really great, and as we'll talk about with the questions this morning, it's going to really be a treasure trove of information and give us lots of great insights into SCN2A and what that means across the lifespan for those who have that gene mutation. So just to start off with the natural history study, where are you up to at this point?
So we have tipped over the 100 participants mark, which is fantastic. And I guess we're really only getting somewhere thanks to the participation of so many families from across the world. I think we're up to about people from about 18 countries across 5 continents. So it's been fantastic to have people from all around the world participate. We have had quite a number of people now do a 1-year follow-up assessment following their original baseline assessment with us, which is great. So we're starting to get some of that longitudinal data. And we are starting to do some data analysis for various aspects of the study. And we presented some work recently as a poster presentations at the International Epilepsy Congress and the Asia-Oceania Epilepsy Congress. And from that, we've got a manuscript in preparation, which hopefully we'll submit in the next couple of months.
That's fantastic. You know, having 100 participants so far in 18 countries, that's just a really broad representation and represents just an amazing data set. So hopefully it's going to give us really great insights. And just to give us a bit more context, what is a natural history study and where does it sit in the broader research landscape if you're trying to sort of map out what happens with a particular gene and then where to go with therapies?
Yeah, absolutely. So, I mean, a natural history study is a study that I guess is describing the sort of clinical features, the symptoms of a condition and how they change and evolve over someone's lifespan, and trying to understand the variability of that, those symptoms and changes between people with the same underlying condition. It's particularly in conditions that are somewhat complex, like SCN2A related disorders, is really important that that's not done in isolation, but that it's also done considering what we call the genotype-phenotype correlations. So how those symptoms vary depending on the impact of the mutation. So probably all heard discussed things like mutations causing gain of function, so the sodium channel to be overactive, or loss of function. And really we can't lump those things together in describing the evolutions because they are in fact different conditions even though they're due to the one gene. So a lot of the time our work is not just focused on the evolution of the condition but also trying to delineate different subgroups so that we can do the natural history in the most useful way. So you're on mute, I think, Dave. Are you while you're on mute, I could just add that one of the important reasons, I guess, for doing the natural history study are really twofold. One, I guess, to be able to give clinicians and families an idea of what to expect for the future, the kind of things that we should preempt or screen for or might be able to treat early, the kind of treatments that seem to work best for conditions, but also as important baseline or historic data for future clinical trials. And that's both for, I guess, guiding which people might be candidates for the trial and what the trial inclusion criteria should be, but also as a sort of, I guess, historic control data set upon which we can measure outcomes. And while a lot of clinical trials are designed such that you've got a control group and sort of, I guess, what we call placebo control, where you're trying to do this really robustly, comparing two different groups at the same time. And in some cases, we can do that for rare disorders, but in others, it becomes quite tricky just because of the number of people. So having that control data set is really important.
Yeah, and I think one of the strengths of this type of research as well is the longitudinal nature of it, because a lot of studies will take a cross-sectional data point and say, right, at this point in time,
yep,
this is what's going on. But then you don't know, was something different a year, 2 years down the track? Does it evolve?
Yeah, absolutely. And we see also, I guess, different subgroups of severity of the conditions as well, and being able to say, well, if your symptoms and sort of level of severity is here, what would we expect the next year or two to hold for you, as opposed to measuring the whole group and seeing a lot more variability. That makes it less useful, I guess, to be able to predict where an individual person may be in a year or two's time. So that's really helpful from that point of view.
Yeah, and then recently there's been a bit of a switch in some of the data that you're looking at to a more quantitative sort of focus.
Yep.
Why? And why is that important?
Yeah, so that is really important for the possibility of future clinical trials and using this as a historic control data set. In order to do that, we really need to be really precise and quantitative and be able to put numbers on things. And that is less important for describing the condition itself. And there are other ways in which we can get natural history data. For example, what was the age at which someone attained particular milestones like sitting or walking, things like that. And it's actually really important that we do that, although we know that that's retrospective and obviously is subject to someone's recall about when that happened or what was documented in the medical records. And that is really important and does help us sort of delineate subgroups and give us information about how things progress over time that is useful without having to wait years to get all of this prospective longitudinal data. But it doesn't quite stand up in the same way in a clinical trial historic control data set, and that's really critical that we start to get that happening now.
Yeah, and you, as we talked about, you've got about 100 participants and you're starting to get manuscripts ready for preparation. So what are then the next steps? Where are you going with the natural history study?
Yep. So I guess, there's a number of things that we want to do. We're continuing to collect the prospective data so that we've got a longer data set over time. We are still interested in recruiting more participants because I guess given this is a sort of complex set of disorders really rather than one, even though we've got 100 participants overall for each subgroup of SCN2A related disorder, there are fewer. And so the more the merrier at this point in terms of helping us have really robust data. The other thing going forward in terms of clinical trials is looking at characterising the variants in the laboratory, trying to determine if things are gain or loss of function, which again is really important for who might be candidates for clinical trials and helping us have the most robust understanding of how the clinical features fit with that. And we hope to be able to characterise the variants of all participants in the natural history study. And so that's a focus at the moment. And then obviously we also have all of the data that we've collected so far, which is a lot. And we will have, we're looking at manuscripts, describing the symptom evolution over time, the age at acquisition of skills, what happens with seizures, what are the best treatments for seizures, and various other symptoms. Particularly in the early onset subgroups of those with seizure onset in the first couple of months of life, there is a really significant degree of variability in the severity of symptoms as children get older. And how do we predict where things might be in a couple of years' time, which is really important to be able to give families an understanding of that, but also to say, well, if we would like to do clinical trials and we would like to treat people as early as possible to be able to get what we think will be the best effect, the earlier we treat, the better is our presumption. How do we determine who is going to need a new and disease-modifying treatment in order to have a good outcome, and whose outcome might actually be okay without that?
And as things progress and there's more and more treatments sort of come into development, and we start to understand things, this really rich data set that you're developing as part of the natural history study, how is that available or able to be utilised by other groups around the world who are also looking at developing treatments or trying to understand SCN2A?
Yep. So I guess there's a number of ways there. So, I mean, firstly is the standard scientific one, that publishing the data means that publishing manuscripts means the information is available to people. We also have a collaboration with the Simons Foundation in the US, and many of you will know does research into a lot of different genetic disorders and has quite a large cohort of people with SCN2A related disorders. And we have a bi-directional data sharing agreement, which they can share their data with us, and we share ours back with them, providing the parents of participants consent to that. That's part of our consent forms. Any data that the Simons Foundation holds is available to researchers who have ethically approved studies. They have a process by which you have to apply, but it is quite widely available to researchers. So that's an avenue whereby people can get access to our data.
Yeah, it's great and really important that data is available, broadly. So it's, and that's important for the community and families to understand that this isn't just sort of held in a silo and only kept for a specific purpose, but we're really building a strong, rich resource that's going to inform research for years ahead in this area.
Yep.
So I'll just go to a couple of specific questions that have been asked. In your initial data that you've been looking at from the natural history study, have you shown any signal or anything about medications that may be beneficial for loss of function and symptoms of spasms or seizures?
Yeah. So might be useful to sort of preface that question just with a little bit of an explanation about what we mean by gain of loss of function, which many of you will know, but in case people don't. So there are different subsets of SCN2A related disorders which differ in their clinical symptoms, and that has a really strong correlation with whether the mutation causes the sodium channel to over or under function. And because some of our medications, particularly for seizures, impact the sodium channels and they actually block the sodium channels, you would anticipate that those medicines might be helpful for some people, but actually potentially make symptoms worse for others. So when we're talking about treatments, it actually is really important that we differentiate those two groups. From a clinical point of view, those with a gain of function mutation almost always have seizures, and they almost always have seizure onset before age 3 months. A small proportion can have seizure onset a bit later, and they are children who usually do quite well, both from a seizure and developmental outcome point of view. But for all intents and purposes, we consider that the gain of function group to be children who have seizure onset before 3 months of age and mostly in the first week of life. Those with loss of function phenotypes, there are, again, a couple of different subgroups within that. Some children have no seizures at all and have either or both of intellectual disability or an autism spectrum disorder. Or they may have seizures, and they can come up, you begin in sort of mid to late infancy, usually as infantile spasms, or they can have seizures that begin later in childhood when there's variable different types of seizures there. So to go specifically to the question of which treatments have benefited those with loss of function disorders, I think the treatments that we have for those conditions, because the sodium channel is already under functioning, blocking it further would make things worse. And certainly that's what we've seen in a number of children. Sometimes seizures really go through the roof with those medicines. Having said that, that's not the case with all people, and there are a few reports where people have benefited from them, and that's something that we actually don't quite understand. We don't know what the explanation is for that. But for a lot of children, they're not great drugs to be on. In contrast, we don't have seizure medicines that really specifically increase the function of sodium channels, because in many cases that would make seizures worse for a lot of different types of epilepsies. So we don't have anything that's quite as specific for seizures in the way that we have for the gain of function mutations. There doesn't seem to be one medicine or treatment in particular that is clearly better than others, but we have seen a number of treatments that have benefited a number of children, and all of these, I guess, come with the caveat that there are observations in small numbers of people only and not robust clinical trials. But if we look at the children with infantile spasms, sort of mid-infancy kind of onset, some children have benefit with steroid medicines, which is usually our first-line treatment for infantile spasms, although in a number of children we've seen that this is transient and the spasms can come back. But for some children, they settle down. Other treatments that have been quite helpful include the ketogenic diet for a number of children. So that's certainly something to consider with your treating doctor, worth having a conversation about whether that might be something to try. A couple of other medications that have helped a number of children have included sodium valproate, which is Epilum in Australia, and Depakote in the US. I'm not quite sure how they refer to it in Europe and Asia, and topiramate or Topamax. In many cases with the medicines I've spoken about, they don't completely stop seizures, although they do in some people. But we've had a number of reports with each of those medicines, and also in the literature of I guess, meaningful seizure reductions in those children. In children with the later onset phenotypes, again, a little bit of a mix and variability in response. But again, sodium valproate seems to come up as a medicine that can be quite helpful in those children as well.
It sounds like for loss of function, you've got a stronger signal or a better feeling of what's not good for loss of function. So what are the things that are not good in loss of function? Yeah,
so again, so sodium channel blocking anti-seizure medicines, and so they include phenytoin, carbamazepine, oxcarbazepine, and lacosamide, which are dilantin, tegretol, trileptal, and Vimpat, are the sodium channel blocking anti-seizure medicines. And again, our observation is that some children's seizures are considerably worse. But there are also reports where there's not much change or even a little bit of improvement. But on the whole, our impression is it's probably better to avoid those medicines.
And then how does that differ for gain of function?
Yep. So gain of function, in contrast, if you've got an over-functioning sodium channel and you block the function of that, then you're bringing the activity of the channel down to closer to the expected level of function. So those medicines can often improve seizures, sometimes completely stop them, but not always. But even if they don't completely stop them, we usually do see benefit in terms of frequency of seizures, and sometimes that's enough benefit to be able to have children well enough to no longer need to be in hospital, which is obviously a meaningful benefit even if things don't stop.
And what about medications for movement disorders in either gain or loss of function?
Yeah. So look, that's actually a really tricky symptom to treat, and we don't have really great answers for that at this point, unfortunately. There are a number of different movement disorders that you can see in SCN2A related conditions. And by movement disorders, we mean conditions with involuntary movements that are not seizures, and they're due to dysfunction in some of the deeper structures in the brain, and with producing movements that children can't control or people can't control. And that differs a little bit from difficulties with motor function, stuff like delayed gain of gross or fine motor milestones or a general clumsiness or things like this. We're talking about involuntary movements. And in some of the children with gain of function mutations, you can see a movement disorder called dystonia. And that usually comes up in children who have more severe impacts overall of their condition in terms of development and seizures. That can be really, really problematic and impact on comfort, ability to do their motor skills, ability to feed, and is really tricky to treat. A number of children that we've seen have had some benefit from Botox injections, and often that's done to specific muscle groups targeting particular problems, because you can't inject things like that everywhere. So it's often done in quite a targeted way to improve one aspect of function, and it can have significant benefits. A lot of the medications are quite variable in their effect. We've had a couple of reports of children having some improvement on gabapentin. In fact, quite a lot of children with the severe early onset phenotypes are on gabapentin, not always for a movement disorder, but we do sometimes use it for symptoms like irritability as well. And so sometimes it's used for other reasons, but there are a few reports of benefit with gabapentin for dystonia. And with most of the other treatments being quite variable, sort of single reports here and there, and sometimes things not working or even being a little bit worse or just incurring potential side effects of the medicine. So unfortunately, it's a really difficult symptom to treat. One thing that we have noticed a little bit is a bit of an inverse relationship between seizure, how good seizure control is, and how bad the movement disorder is. And sometimes we find that the dystonia is worse when seizures are better. We don't really understand why that is. Sometimes it comes up at a little bit of a later age where, not sort of in the first few months of life, but maybe at a year or two of age, it becomes much more apparent, and maybe, maybe it's purely an age-related thing, or maybe it's just that when someone's having a lot of seizures, they're really quite sleepy and sedated often. And in order to have a movement disorder and have the problems associated with movement disorder, really you see them when someone's awake, and it may be related to that as well. But obviously that's a real trade-off in terms of which symptom is more problematic. Sometimes we find that the medications used for seizures sometimes can seem to make movement disorder symptoms a little bit better or a little bit worse. They're not great treatments in terms of making things better, but sometimes people notice a little bit of benefit, but actually sometimes people notice they're a bit worse as well. And so sometimes tweaking the seizure medicines can have a little bit of impact. But overall, I'd say we really don't have great treatments, and that hopefully would be something that novel treatments can address. Within the gain of function group as well, some of the children whose level of severity is a little bit more sort of intermediate have a movement disorder called episodic ataxia, which basically means that much of the time they don't have involuntary movements, but then they have episodes lasting hours or days where they are ataxic, so unsteady, not able to sit or stand or walk in the way they usually would. Sometimes it's accompanied by things like vomiting or funny eye movements and dizziness, which is quite an unpleasant symptom. There was a paper out about 18 months ago looking at a cohort of children with SCN2A related episodic ataxia and reporting some benefit with a medicine called acetazolamide or Diamox in a proportion of patients. That would be something to try for that. And so that's within the gain of function group. Those are the main movement disorders. Within the loss of function group, some of the children, mainly the children who have the infantile spasms presentations of seizures, can have a movement disorder called chorioathetosis. which is sort of excess movements that have a arriving kind of quality to them. They're probably less problematic than the dystonia in the degree of discomfort and those kind of problems that they exhibit. They can have a major impact on someone's ability to do gross and fine motor skills, and so they really can impact life and quality of life. Again, we don't have great treatments for those, and we haven't seen of the children we've seen with that, we've not really seen medications that have particularly helped. There are a few reports of people having some benefit with changing their seizure medicines, and so sometimes there's a couple of reports of people whose movement disorders have improved after coming off a medicine called vigabatrin. Vigabatrin's actually, their relationship of vigabatrin with a side effect of movement disorders is actually not SCN2A specific. It's something that is known. But we heard a couple of people say things were better after they stopped that. But on the whole, no great treatments, unfortunately. Again, hopefully disease-modifying treatments will address that.
Yeah, and then this is more drawing on your clinical experience, of a lot of the treatments you've talked about, be it for seizures or be it for movement disorders, what are some of your long-term concerns about use of those medications? Because again, we're often in the here and now. It's like, let's start it, let's get control. Yep. But if we're trying to then maintain that control, what are the things you worry about in the longer term?
Yeah, so I think, in that kind of context, we'd sort of probably separate out seizure control from other side effects. Oftentimes from I guess to start with this gain of function presentations, oftentimes when you hit on a seizure medicine that works, you tend to get continued benefit from that medicine, although sometimes doses need to be adjusted for a weight. As you get bigger, you need more medication. But we tend to see that those medicines still have benefit. In some children with sorry, I should put my pen down, early onset conditions of more sort of intermediate severity, sometimes they can have seizures that come up into the primary school age years or preschool and primary school age years that are a little bit different to the seizures they had during infancy, and sometimes seem to benefit more from different seizure medicines other than sodium channel blocking drugs. So sometimes we do need to think, do we actually need to change our treatment tack a little bit and also use something with a different mechanism of action? But for the most part, it's the sodium channel blocking drugs that work and have ongoing benefit. One of the issues with seizure medicines, which we're very cognizant of and which families will be very well aware of, is the side effects that can happen, and that can include things like sedation and just sort of, I guess, being sleepy or being a bit dopey, not being as alert and interactive. And we don't want children to be overly sedated. We don't want to be causing harm or impacting on things, because the developmental impacts of these conditions can be significant, and we don't want to be adding to that. Generally, I guess an important thing is to not be on medicines that aren't helping. And sometimes when children are having a lot of seizures and there's a lot of medication changes in a short space of time, sometimes medicines will be added. But it's always important to consider, is everything that a child's on actually helping, or should we come off some medicines? And certainly, there's many reports in SCN2A and others. As clinicians, we see it all the time, that parents will say, look, my child was brighter when they came off this medicine, and they were doing more and interacting, and life was better. And I think as clinicians, we need to be really cognizant of that and try and rationalise the medicines as much as possible. It's very difficult to quantify the impact of that kind of thing, but I think we do need to be mindful of not using treatments that probably aren't giving benefit. As far as sodium channel blockers go in the long term, look, some of these drugs have been around for many, many years, and we don't have particular concerns of sort of accruing impacts on organs for at least some of these medicines. For some of the medicines, we know that side effects can become more apparent over time. Things like with Dilantin, the gum overgrowth, for example, and things like impacts of bone health and that kind of thing. But from the point of view of sort of kidneys and lungs and that kind of thing, generally we're not worried about that kind of thing. And overall, we're always considering things in terms of risk benefit, and maintaining seizure control is an important thing. As far as loss of function goes, look, things are, I guess, are really similar principles. I think one of the things that's really quite noticeable is the impacts of sedation and things by being on too many medicines. And it's sort of, I think that would be probably the main point to make there.
Yeah, and do any of the medications have an impact on pubertal development?
Probably not the medications in and of themselves, but I think there was a question about precocious puberty.
So that sort of leads into
puberty.
So if it's not the medications, does SCN2A itself have an impact on pubertal development?
Yeah, so look, it's hard to know. We do know that early puberty can be a feature in lots of different neurodevelopmental disorders. We see that quite a bit in children with delays in development, intellectual disability, other neurologic symptoms. So it's certainly not something that's specific to SCN2A by any means. We've seen it in a number of children, but not, I would say, not in a high proportion, although with the caveat that many children are younger than we would expect even for early puberty. And my impression is I don't think it's more common than in children with other neurodevelopmental disorders, but I think we need some more data to really firm that up.
Okay, so we talked a little bit about medications. Just to come back to one of the non-drug strategies, you mentioned the ketogenic diet.
Yep.
What's the role for that in terms of both, potentially seizures and sort of general well-being of kids with SCN2A?
Yeah, so I think ketogenic diet can be helpful for seizures. We've seen it help a number of children with a loss of function disorders, so chiefly the spasms presentations. One of the things that can be helpful in that respect is that sometimes it also allows you to come off the seizure medicines. And the ketogenic diet is not sedating in and of itself. And if you can be on fewer medicines, then maybe that's a positive thing. So I would see that as one of the potential benefits if it works. But it is really important to note that it's a medical treatment. It's a diet that needs medical supervision and needs monitoring for potential side effects in the long term, which are not trivial. It's also not the most straightforward thing to do if you're also feeding other members of your family, because you do need different treatments, and it can be difficult particularly in an older child or a child who feeds orally as opposed to having a gastrostomy tube, in that the tastes may not be ideal, and in children with difficult behaviours or oral sensitivities or things like that, that can really be very difficult. But where it's doable and you see some impacts on seizures, then then that can be a really good thing. But there's situations where it's doable and some where it's just not.
Thank you very much for that. So just I'll shift away from some of that treatment. We'll come back a bit, talk a bit more about some of the clinical trial, the natural history information that you're getting. In what you're looking at, if you're finding predictors of long-term severity or things you can sort of look at one point that are going to give you some indication of what the future holds?
Yep. Yes. The answer is yes to that. And I think we see that most strongly in the gain of function phenotypes, so the early onset seizures, where the severity in the longer term can range from a condition we consider to be benign in the sense that seizures remit and development is normal, through to very severe in the context of having an ongoing treatment-resistant epilepsy with really severe developmental impacts and movement disorders, and then some people with more intermediate severity of symptoms. And the early clinical features present in the first month or so of life in those children do reasonably strongly differentiate those who are likely to have a severe outcome from those who will be less severe, as either benign or more intermediate. I guess we're talking about clinical features there. So, things like were there seizures in utero, things like what's seizure control like early on and how frequent are seizures, things like what is the child like in themselves when they're not having seizures? Are they able to suck and feed? What's their tone like? Are they floppy, or are they is their tone more as expected for age? There's a number of features that span those kind of clinical symptoms, which do seem to be quite predictive, and hopefully we'll have some robust data out of the study from that point of view. As far as the later onset phenotypes go, our impression to date is that with the children with the sort of mid to late infancy, infantile spasms type presentations, the overall severity seems to be a bit more homogeneous in the sense that we don't see the same spectrum of severity. Things are a bit more similar in the long term, obviously, with a little bit of variation, but a lot less so. But in some children, we see seizures sometimes settle down or improve, but in many, they continue to have ongoing seizures. And development obviously impacted as well. And I think the data coming out of the study will give us a better idea of where a lot of these children's sort of how they progress and where things end up. As far as those children with no seizures or with seizures that begin later, again, the severity is a little bit variable, but probably closer to in terms of how homogeneous it is probably closer to the mid-infancy than the really big spectrum we see with the early onset children. In fact, a lot of the children with childhood onset seizures, in the first few years of life, their symptoms and their course really parallels that of the children who never have seizures, where we sort of see some mild delays in the first year of life, sometimes with no delays noticed even until sort of the sort of 9 months or so. Delays become quite apparent at the sort of 12 to 18 month mark when language doesn't develop in the way you would expect. Some children having a significant regression of language and social skills then, and then very slow progression after that with relatively good motor skills. That pattern is quite similar across those two groups, and then some children get seizures and some don't. And we don't really know why that is. Hopefully we'll be able to tease that out a little bit further in time, because I think in children whose initial symptoms and reason that they're diagnosed is because of their developmental concerns rather than seizures, one really strong worry for parents is, will their child have seizures? At the moment, we're not able to predict that particularly well.
Yeah. And in some of the animal models, at least or at a biological level, there's this change in the sodium channel that the SCN2A gene affects seems to be really important early in life.
Yep.
And then less important as development progresses.
Yep.
Have you seen that in some of the work you're getting from the natural history study, that there's a vulnerability and symptoms early in life that then either change or are different a bit later on?
Yeah, yes, the answer is yes. I guess what you're referring to is so SCN2A is the gene for the NAV1.2 channel, which is one of the sodium channels in excitatory neurons in the brain. I guess the so-called accelerators rather than the inhibitory neurons, which are the brakes. And the NAV1.2 protein is what's really important in early life. And then over the first couple of years of life, the SCN8A gene, which produces the NAV1.6, confusingly not 1.8 channel, which is also in excitatory cells and becomes the dominant sodium channel then over the first couple of years of life. In the children with the gain of function or early onset phenotypes whose seizures settle down, the timing of that parallels the change in the which is the predominant sodium channel, and it may be that that's the reason why things settle down around that age. We do in children with gain of function phenotypes whose symptoms are a bit more severe, but not necessarily at the most severe level, also find that seizures settle down around that time, may stop or may just become better controlled, sort of 1 or 2 year age mark. And again, that probably reflects that, certainly our hypothesis. As far as the children with the more severe early onset phenotypes and those with later onset phenotypes, that kind of age-related changes around those age are probably less marked than in other children, although we do sometimes see some changes.
Yeah. And in terms of the phenotyping, we've got that sort of bigger structure of gain of function versus loss of function, with that sort of early onset versus a bit later onset, more of an autism, less of a seizure sort of presentation. In the work that you're doing, talk to us about a framework for the subsets of that. So you're sort of finding you're drilling down into sort of smaller categories within each of those larger categories?
Yep. Yeah, so within the gain of function or early onset groups, so the sort of, I guess, benign, intermediate and severe groups, the children with more benign seizure types, so tend to often have less frequent seizures to start with, although still often multiple daily, but generally not multiple hourly for long periods of time. They tend to have seizures that are treatment responsive, although they may not be responsive to non-sodium channel blocking drugs. So having a period of treatment resistance early if you're not using a drug that's specifically helping the impact, we certainly do see that in some children whose outcome is later positive. What we see in those children is that seizures remit and children can come off seizure medicines sort of in early childhood, usually is when we would expect the tendency to have reduced, and that development progresses as expected. Sometimes early on, there can be sort of treatment-related sedation and things be a little bit late to start with, but we tend to see good outcomes in those children. So we refer to that as a benign phenotype, even though it's not benign to have a lot of seizures in the neonatal period. The outcome's good, and that's the reason for that terminology. Comes about from the initial description of SCN2A related disorders being a type of epilepsy known as benign familial neonatal infantile seizures, and that can run in families. They can be inherited mutations, and one of the reasons they can run in families is because people do well and in adult life lead normal life and have their own children. And we now don't really refer to that as benign, but more as self-limited. As far as the severe group goes, and I'll come back to the intermediate, the severe group, if most, but not all people have treatment-resistant seizures. So seizures will improve with sodium channel blocking drugs, but may not necessarily go away. And they're much more likely to have a seizure tendency that goes beyond age 2. Again, that's not absolute, but the proportion is a lot higher. Some children will develop infantile spasms or other generalized seizure types, things like myoclonic seizures or tonic seizures, or even reflex tonic seizures. Sometimes things like noise or larger things can trigger seizures. We tend to only see those generalized seizure types in the more severely impacted children. Development is usually delayed from the start and continues to be really significantly behind, and ultimately with an intellectual disability, although in many instances it's actually quite difficult, I guess, to determine the sort of level of cognitive function, because the motor impacts are often really quite severe, and being able to communicate and do things where it does, particularly in young children, relies a lot on your motor skills as well. So it can be difficult, and sometimes it becomes clear as children get older they actually understand a lot more than we might have realised when they were younger. So, but yeah, so there's a little bit of variability there, and some children are actually able to communicate really well with things like communication devices. And movement disorders, particularly dystonia, are really problematic in these children. Often that becomes more apparent at the sort of 1-2 year age mark. Many children will have difficulty with feeding and may need gastrostomy feeding. And sometimes we can see other things crop up as problems. Something that's not really in the literature, but which we've seen a number of times in these children, is urinary retention. So having periods of time where you're not able to pass urine, which can be uncomfortable and might need a catheter to help with that. So it's certainly something we've seen in a number of children. As far as the more intermediate severity group goes, I think there's sort of splitting these up into three groups. I think really sort of comes back to the question of you, someone who likes to split things into groups or lump everybody together. And I think people have got different ideas about that. I think the groups at both ends are really quite distinct. Group in the middle is a little bit more of a question of, is this a gradient of severity? And certainly it's not a totally homogeneous group, but for the purposes of sort of describing these things, it is useful. Most children with these with that phenotype 10, most of the time seizures do settle down at around the age 2-ish mark. Some children have some ongoing seizures, and some children have a period of time without seizures, and then they recur and can be a problem for a couple of years and often settle down again after that. But seizure control tends to be better than in the other children. There's real variability in development. It can be normal, or there can be an intellectual disability or autism. One of the things when we look at the sort of different levels of function, most of the children, in fact, really almost all the children in the intermediate group are ambulant, so they're able to walk. They may attain that milestone later than expected, but they can walk, and that is a difference between that and the more severe group, with sort of language and cognitive function being more variable. Some children have movement disorders, although generally not dystonia. Probably the most predominant movement disorder we see in this group is the episodic ataxias that I mentioned before. So I do use the sort of intermediate grouping, even though we know it varies. And I think there's a few things there that we can pick out that are quite consistent, and a couple of things that are more variable there. Generally, I would consider the intermediate group to mean that you have some symptoms beyond age 2. And that may mean you have ongoing seizures, that may mean you have a movement disorder, that may mean you have impacts on development. You might have one of those or all three.
And then if you're looking at typing SCN2A more by genotype, so particular gene variations, we often see, though, that translates to different clinical presentations. How does that happen? Someone's got similar gene variant, but the clinical presentation is different.
Yeah, absolutely. So one thing to say is that so far we see, even if someone has exactly the same gene change, exactly the same spelling mistake in a gene, we see that phenotypes, the sets of symptoms, split nicely between those with a gain of function phenotype or loss of function phenotype. So we don't see one mutation causing neonatal seizures in one person and the West syndrome presentation, infantile spasms presentation as your first seizure type. We don't see that. What we do see is everybody sort of fits within the same overall group, but that the severity can vary. And to give you an example there is in the early onset or gain of function ones with all of the recurrent variants, so variants that have been reported in 2 or more people, about 40% of those variants were seen in individuals with different levels of severity. And sometimes that ranged from some individuals with a particular variant having a very profoundly severe phenotype, and others having a completely sort of benign course to their condition. And that's, I guess, something that's tricky to understand, really, because, why would that be? There's lots of potential reasons for that, and it's still not well understood. People are trying to characterize that. It may be that there are other genes that are modifying the impact of the SCN2A related condition, and that the way other channels are working can either compensate or not compensate for the impact of the SCN2A related thing. It may also be aspects of the SCN2A change itself, not the mutation type, but how much that the gene with the abnormal copy of SCN2A is expressed. If for example a lot of your brain cells were using more of the unaffected copy, because all our genes come in 2 copies and only one of them is mutated. If your body was using more of the normal copy, you may have fewer symptoms. So there's lots of potential reasons, and we actually don't understand why that is.
Great. Thanks very much for all of that, Katherine. I'll hand back to Chris to close out the webinar.
Well, thanks, David, for running that session for us. And Katherine, just the conversation that you've just shared with us by answering those questions really shows the importance of participation in the natural history study and the rich data that you're getting from it. Some really brilliant insights. I was listening intently to your answers, and it's really great for families to see that their participation in the natural history study is really leading to a much richer understanding of SCN2A altogether. I just wanted to, well, finally thank you, but also just give you the opportunity if there was anything you wanted to say that we haven't covered, a call to action to the SCN2A community. I'll just leave it with you to close, but thank you for your time.
Nice. Thank you, Chris, and thank you, Dave. Probably a couple of quick things to say. I think a lot of my answers today probably focused a bit more on the gain of function and early onset conditions rather than the later onset or loss of function conditions, which I think is probably partly because we understand the early onset ones a little bit more. But I just wanted to reassure people that we are working really hard on understanding the other conditions better. We've got a lot of active stuff going on there, so watch this space on that front. I want to say thank you to all of our participants. The participation, I guess, takes time and takes a lot of emotional energy as well, revisiting all of these things, and we really appreciate all of your participation and support of the study, and hope that this will change things for the better in the future. And the other thing I would say is some of you will have already been contacted by us, and some soon, to get to do the quantitative aspects of the study, so including the Vineland developmental assessment, which is done as an online survey, where that's the quantitative bits of the study that we're trying to get going, and we're really keen to be able to do that, to sort of enrich the quantitative data set to move things towards trials. So definitely a call to action when we get in touch with you. If you could do that, that would be brilliant. Thank you.
Thank you, Katherine, and thank you everyone for joining us. We will have this recording available to anyone who would like to watch it in the future. And thank you.
You're welcome.
Thanks, everyone.
Thank you.